Hi, everyone. Thank you for taking the time to learn about my research project tonight. My name is Adelina Lipton, and my research focused on analyzing raptor tissues to identify organochlorine pesticide residue in the southeastern US. And before I get started, I would like to thank Queen's University for facilitating this research, the Carolina Raptor Center for providing raptor tissues, and of course, the Mecklenburg Audubon Society and the Nobel Fellowship for providing funding. So to start off, what are organochlorine pesticides? Organochlorines are synthetic compounds used primarily in the agricultural industry for their insecticidal properties. And organochlorines, including DDT, are highly toxic and persistent and accumulate within organisms. Most have been banned since the 1970s to 80s due to the recognition of the harmful effects they have on human and environmental health. As for the effects on raptors, organochlorines have been known to cause reproductive issues and developmental defects, including embryo mortality, deformities, eggshell thinning, and edema, as well as neurological dysfunction and motor skill impairment. As apex predators, raptors biomagnify contaminants, putting them at a substantially greater risk of these compounds. These complications can drastically impact a raptor's ability to survive in the wild and reproduce, which is evident in the historical decline in raptor species, which are a direct result of organochlorines. An example of this is the 1975 New Jersey osprey population, which was at 66 pairs, which starkly contrasts the historic population of 350 to 450 pairs. Following the ban, levels have decreased and raptor populations have steadily recovered. Raptors provide valuable ecosystem services, including disease control and waste removal, and an example of this is that declines in vultures could cause more exchange between mammalian scavengers at carcasses, which facilitates disease transmission. Raptors are able to quickly respond by their ability to fly. In addition, while flying throughout their home range, raptors transport nutrients from one place to another in both terrestrial and aquatic ecosystems, which is vital for plant growth in areas with limited nutrients. As apex predators, raptors are especially useful to study to determine ecosystem health, as they act as bioindicators of environmental conditions that can also impact humans. And an example of this is the canary in the coal mine. The Carolina Raptor Center is a nonprofit raptor rehabilitation facility located in the Charlotte region. And despite the general trend of organochlorine residues decreasing in the environment, since 2016, they have received raptors with neurological dysfunction, which is a sign of disease and spinal injury, but also of pesticide toxicity. And these raptors died regardless of treatment within 24 hours of arrival. Following extensive investigation, some of these fatalities appear to be caused by extremely high levels of organochlorines. Provided by the Carolina Raptor Center, between 2016 to 2018, tissues from 41 birds with neurological symptoms were sent off for chemical analysis. Of these birds, 7 were found to have extremely high organochlorine residues in either their liver or brain tissues. One red-shouldered hawk was found to have 59.2 parts per million of dieldrin and 6.2 parts per million of heptachloroxychlordane in its brain. For reference, the lethal thresholds of these compounds are 4 parts per million of dieldrin and 5 parts per million of heptachloroxychlordane in the brain. Another example is a red-shouldered hawk that was found to have 705 parts per million of dieldrin in its liver. And it is important to note that this is a liver sample, so it can't be compared to the lethal threshold for the brain, but it is extremely concerning nonetheless. As for potential hypotheses, one goal of this study was to determine if exposure is limited to migratory raptors. This is because if it is limited to potentially migratory raptors, they could have been coming into contact with organochlorines in certain countries in South America that have not yet banned all organochlorines. And this could happen by them storing the organochlorines in their fat and then actually metabolizing it when flying back to the US, which could be why they're presenting in the US. If this issue is not limited to migratory raptors, this could be an issue of purposeful poisoning or illegal use of organochlorines. And one thing to note is that I will be referring to these raptors as either potentially migratory or non-migratory, because although potentially migratories do possess the ability to migrate, not all choose to do so, so we have no way of knowing for sure whether they did migrate or not. And another thing to note is that there could be other hypotheses. These are just the ones that we determined to be the most likely. Raptors were acquired from the Carolina Raptor Center. We collected 40 liver and brain tissue samples from 27 raptors. These raptors were not selected based on cause of death or symptomology, as one goal of the study was to determine how widespread this issue is outside of just those presenting with symptoms. For example, the cause of death might have been determined to be hit by a car. That raptor could have been hit by a car because it was flying abnormally due to organochlorine exposure. So that is why we decided to choose them non-specific to cause of death. 24 potentially migratory raptor tissues were collected and 16 non-migratory tissue samples. And as you can see in this graph, you can see the species, whether they are potentially migratory or not, as well as the quantity of tissue samples collected. And I'll go down the list to explain the acronyms. So BDOW is barred owl, BLVU is black vulture, COHA is Cooper's hawk, GHOW is great horned owl, OSPR is osprey, RSHA is red-shouldered hawk, and RTHA is red-tailed hawk. As for the experimental procedure, each of the selected raptors was dissected by performing necropsies in order to access their liver and brain tissues. Tissue samples were homogenized and dehydrated using a mortar and pestle followed by sodium sulfate. Non-target compounds were removed using a catcher's kit, which stands for quick, easy, cheap, effective, rugged, and safe, and it is just that. It is an extremely effective method at cleaning up samples. Following this, samples were sent to the Mississippi State Chemical Lab, where a GCMS, or gas chromatography mass spectrometry instrument, was used in order to verify and quantify organochlorine residues. And 17 organochlorine pesticides and metabolites were targeted for analysis. To assess our extraction methods, we first analyzed 3 blank chicken liver samples by putting TCMX DCBP in them, and this is essentially a spiking solution. And we put this in so that we can make sure that we are getting all of it out that we are putting in. So we put this into every sample to assess our recovery rates so that we can essentially tell that if we are getting the exact amount of TCMX DCBP that we put in on any given sample, then we can make the assumption that we are also getting the right amount out of the actual pesticides that are targeted for analysis. Acceptable recovery rates are within 80 to 120%, so as you can see, our recovery rates fell well within that range. We statistically compared the mean values of each organochlorine pesticide using a one-direction, two-sample t-test, analyzing concentrations of liver and brain samples separately. A correlation between brain and liver tissue concentrations was determined using the correlation coefficient. 5 out of 17 organochlorine compounds were detected. However, the only organochlorine consistently detected was 4,4-DDE, which is a breakdown product of DDT. So this is the compound that we chose to run analyses on. 11 raptors were included to determine the relationship between liver and brain samples. Liver and brain tissues are significantly positively correlated in regards to concentrations of 4,4-DDE. And as you can see in the graph, with liver on the x-axis and brain on the y-axis, They have a positive and linear correlation. This graph for the liver concentrations of 4,4-DDE included 14 potentially migratory and 12 non-migratory tissues. No significant relationship or difference was observed between liver concentrations of 4,4-DDE between potentially migratory and non-migratory raptors. As you can see, the mean for potentially migratory raptors was 0.755 micrograms per gram, whereas for non-migratory raptors, the mean was 0.645 micrograms per gram. As you can see with potentially migratory raptors on the left and non-migratory on the right, there was one outlier in the dataset for potentially migratory raptors. However, overall, there were no significant differences. This graph for brain concentrations of 4,4-DDE included 8 potentially migratory raptors and 4 non-migratory raptors. Following the same trend as liver, no significant relationship or difference was observed between brain concentrations of 4,4-DDE between potentially migratory and non-migratory raptors. As you can see, the mean for potentially migratory raptors was 0.0116 micrograms per gram, and for non-migratory raptors, the mean was 0.016 micrograms per gram. As seen on the left, potentially migratory, and on the right, non-migratory raptors, potentially migratory raptors did have an outlier, but there was no significant difference. As for the results, there is a significant positive correlation between liver and brain concentrations. This is extremely useful to know because liver is more commonly sampled. However, the existing lethal thresholds are for the brain. There is limited data on lethal thresholds of organochlorine compounds in the liver. Therefore, future studies with a larger sample size and more diverse sample pool could examine whether liver levels could be used to predict brain levels, which could then determine exposure significance. As for potentially migratory versus non-migratory, organochlorine exposure does not appear to be a widespread issue that raptor species face, as tissues were selected at random and non-specific to cause of death or symptoms. And no statistically significant patterns were identified. Relatively low levels were detected regardless of migration patterns. And although steadily declining, 4,4-DDE is shown to be the most abundant and persistent organochlorine, which is likely the reason why it was the most consistently detected throughout the tissue samples. As for implications for the Carolina Raptor Center, these findings are valuable to make sense of the 7 raptors that they received with extremely high concentrations in their liver and brain. This does not appear to be a common issue anymore, as supported by current research as well as my findings. Because of this, the source of the extremely high concentrations could be local and an incident of purposeful poisoning due to the unreasonably and inexplicably high levels, and because no correlation was found between migratory raptors. Purposeful poisoning of raptors, which is most often done by farmers to stop them from preying upon livestock, is a relatively common occurrence, although rarely documented and acknowledged. Therefore, it is a viable possibility. Insecticides, including organochlorines, are among the most frequently involved in pesticides in cases of animal poisonings. However, much more extensive research and investigation would be needed to confirm the possibility of deliberate poisoning. It is important to note that contamination could be due to a combination of various sources mentioned or those not yet identified. We can, however, likely rule out foreign countries that have not yet banned organochlorines as the sole contamination source, as organochlorine toxicity is not limited to potentially migratory raptors. Future studies should be conducted with a larger sample size to determine the extent of the organochlorine issue, as well as the relationship between liver and brain lethal thresholds. Additionally, we should study concentrations in raptors exhibiting symptoms of insecticide toxicity prior to death to see whether it was likely the cause of death and to gauge the level of exposure. We should also look at the current most commonly used pesticides that are still legal to determine exposure and impact on raptors. Organochlorines have devastated raptor populations, therefore the proactive monitoring of these compounds is necessary to protect them and continue the trend of the decrease in residues in the environment. Here's my works cited. And thank you for listening and learning more about my research. And I would like to reiterate how vital the Mecklenburg Audubon Society funding was to complete this research, and I am truly so honored to have been awarded. And now, does anyone have any questions for me?